Welcome to EPG Pathshala. I am Dr. Preeti Khandelwal. I work as Assistant Professor in Sri Venkateshwara College. The topic of my lecture is Bioenergetics. In our today's lecture, we will talk about the following aspects: definition of bioenergetics, adenosine triphosphate, that is ATP, proton motive force, aerobic oxidation of glucose and fatty acids, glycolysis, allosteric regulation of glucose metabolism, fermentation, anaerobic metabolism of glucose, oxidative decarboxylation of pyruvate, citric acid cycle, malate aspartate shuttle system, electron transport system, structure of ATP synthase or F0F1 complex, binding change model, transport of ATP through inner mitochondrial membrane, fat metabolism, occurrence of oxidation of fatty acids in mitochondria, and last, summary of the lecture. So, what is bioenergetics? Bioenergetics is an extensive discipline of biochemistry that deals with energy transformation with living organisms and the efficiency of energy transfers among various beings. All living creatures use energy, but in different means. ATP, that is adenosine 5' diphosphate, acts as biologically universal intracellular chemical carrier of energy. It has main role in storage of free energy within the cell. ATP is synthesized from ADP, an inorganic phosphate, and the energy released is delta G is equal to 7.3 kilocalorie per mole. The hydrolysis of terminal phosphoanhydride bonds present in ATP results in release of large amount of free energy. These phosphate bonds are known as high energy bonds. The energy to drive ATP synthesis from ADP is produced from two processes. One is aerobic oxidation that takes place in mitochondria in all the eukaryotic cell. Another is photosynthesis, which occurs in chloroplast, in leaves of plants and single cell organisms like cyanobacteria. In aerobic oxidation, carbohydrates, mainly glucose and fatty acids, are converted into carbon dioxide and water, and the energy is released in the form of ATP. This is the main source of energy in animal cells and most other non-photosynthetic cells. In both eukaryotes and prokaryotes, the initial step of glucose metabolism take place in cytosol and do not require oxygen. The final steps are oxygen dependent and produce majority of the ATP. In eukaryotic organisms, the later steps of aerobic oxidation takes place in the mitochondria, while in prokaryotes, which are devoid of mitochondria, the final reaction occur on the plasma membrane. The initial steps of fatty acid metabolism occur in peroxisomes, and the final stages with the production of ATP occur in the mitochondria in most eukaryotic cells. In photosynthesis, the energy of sunlight is utilized to generate the chemical energy of phosphoanhydride bond in ATP and reserved in the chemical bonds of carbohydrates, primarily sucrose and starch. Oxygen is also released during photosynthesis. In plant cells and eukaryotic single cell algae, photosynthesis takes place in the chloroplast. Several prokaryotes lack chloroplast but are capable to carry out photosynthesis by a mechanism similar to that in chloroplast. The oxygen produced during the photosynthesis is source of virtually all the oxygen in the air, and the carbohydrates produced are the definitive energy source for virtually all for virtually all non-photosynthetic organisms. Proton motive force. Apparently, photosynthesis and aerobic oxidation appear to have little in common, but a revolutionary finding in cell biology reveals that bacteria, mitochondria and chloroplast all exploit the same or very nearly identical process termed chemiosmosis or chemiosmotic coupling to produce ATP from ADP and inorganic phosphate. The instant energy sources that facilitates ATP synthesis are the transmembrane proton concentration gradient and electric potential, that is voltage gradient, collectively known as proton motive force or abbreviated as PMF. The proton motive force is created by stepwise movement of electrons from higher to lower energy levels via membrane-bound electron carriers. In mitochondria and non-photosynthetic bacterial cells, electrons from NADH formed by metabolism of sugars, fatty acids and other substances are transported to oxygen, which is terminal electron acceptor. In the thylakoid membrane of chloroplast, energy absorbed from light strips electrons from water, energy absorbed from light strips electron from water forming oxygen and expedites their movement to other electron carriers, particularly NADP+. Ultimately, these electrons are given to CO2 to produce carbohydrates. All these systems, though, comprise some similar carriers that link electron transport to the pumping of proton, always from the cytosolic phase to the exoplasmic phase of the membrane, thus generating the PMF. Aerobic oxidation of glucose and fatty acids. This mechanism involves the combustion of energy sources such as carbohydrates, sugars and hydrocarbons, that is fatty acids, into carbon dioxide and water with the release of large amount of energy in the form of ATP. The complete aerobic oxidation of one molecule of glucose produces 6 molecules of CO2 and 36 molecules of ATP. The overall reaction is C6H12O6 combines with 6 molecules of oxygen, 36 inorganic phosphate, 36 ADP, 36 hydrogen, which gives rise to 6 CO2, 36 ATP and 42 water molecules. In eukaryotes, glucose oxidation takes place in 4 stages. Stage 1 is glycolysis. It occurs in cytoplasm of cell and involves the conversion of 1 molecule of 6 carbon glucose into 2 molecules of 3 carbon pyruvate. Stage 2 is citric acid cycle, which takes place in the mitochondrial matrix and involves the oxidation of 2 molecules of pyruvate into CO2, then electron transport that takes place at inner mitochondrial membrane and generates proton motive force. Last stage is synthesis of ATP, which occurs at F0, F1 particles present at inner mitochondrial membranes and involves in oxidative phosphorylation. Now, the glycolysis. Glycolysis is the initial step of cellular respiration. It takes place in the cytosol and can be occurred in the absence of oxygen. It is common to virtually all cells and does not require oxygen. So, it is the utmost source of metabolic energy in anaerobic organism. However, in aerobic cells, it is only the initial step of glucose metabolism. The scheme of the glycolysis was given by Embedon, Meyerhoff and Parnas. So, it is also known as EMP pathway. The initial reaction of this pathway are energy consuming. 2 ATPs are utilized for the phosphorylation of the glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6-biphosphate. These reactions are catalyzed by hexokinase and phosphofructokinase respectively. Allosteric regulation of glucose metabolism. There are 3 enzymes which allosterically control the complete glycolytic pathway. Phosphofructokinase is principal rate limiting enzyme whose activity is inhibited by high amount of ATP and activated by AMP. This leads to accumulation of glucose 6-phosphate which act as inhibitor to hexokinase. Therefore, when the cell has ample amount of energy of ATP, the glucose catabolism is inhibited. Pyruvate kinase is also inhibited by ATP. These 3 glycolytic enzymes catalyze allosteric reaction with high negative delta G naught value that are predominantly irreversible under normal condition. The further control is employed by glyceraldehyde 3-phosphate dehydrogenase, catalyzes the reduction of NAD plus to NADH. If NADH amount is increased in cytosol, mitochondrial oxidation slows down. In this pathway, the molecules of ATP are generated by 2 ways: substrate level phosphorylation, direct transfer of phosphate group from substrate to ADP. It is catalyzed by water soluble enzymes, whereas membrane and ion gradients are not required. It ensues twice during the pathway. The first reaction is catalyzed by phosphoglycerate kinase, which transfer the high energy phosphate group of 1,3-bisphosphoglycerate to ADP, generating 3-phosphoglycerate and ATP. The second reaction involves transferring of high energy phosphate group in phosphoenol pyruvate. Transferring of high energy The second reaction involves transferring of high energy phosphate group in phosphoenol pyruvate, which is PEP, to ADP, yielding pyruvate and ATP. This reaction is catalyzed by pyruvate kinase and strongly exergonic. Oxidation of NADH produced during glycolysis to NAD+. In this reaction, 5,3-phosphoglyceraldehyde is converted to 1,3-bisphosphoglyceric acid and the reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. NAD+ is reduced to NADH and later it generates ATP by oxidative phosphorylation in mitochondria. Each molecule of NADH gives rise to 3 molecules of ATP in mitochondria. In this pathway, 1 glucose yields 2 molecules of triose phosphate and 4 ATP molecules, in which 2 molecules of ATP are consumed initially. In addition, 3 ATP molecules are generated from oxidation of each 2 molecules of NADH produced. Therefore, net gain of 8 ATP molecules and 2 molecules of pyruvate occur in glycolysis. Fermentation, the anaerobic metabolism of glucose. In the absence of oxygen, pyruvate formed from glycolysis converts into 1 molecule of ethanol or lactic acid and 1 molecule of carbon dioxide. Pyruvic acid releases 1 molecule of carbon dioxide to produce acetaldehyde. The acetaldehyde reoxidizes NADH and is itself reduced to ethanol. These reactions are catalyzed by pyruvic acid decarboxylase and alcohol dehydrogenase respectively. This mechanism of anaerobic metabolism of glucose is known as fermentation. It is the source of wine and beer manufacture. During prolonged contraction like exercise of mammalian skeletal muscle cells, oxygen becomes limiting. Hence, glucose cannot be metabolized completely to carbon dioxide and water. In this hypoxic state, muscle cells convert each glucose molecule to 2 molecules of lactic acid with the net production of 2 ATP molecules. The lactic acid causes muscle and joint pains. It is mostly secreted into the blood. Some enters into the liver, whereas it is reoxidized to pyruvate and either further metabolized to carbon dioxide and converted to glucose. In the heart, considerable amount of lactate is metabolized to CO2, which is highly perfused by blood and can continue aerobic metabolism occasionally. When exercising, skeletal muscle cells secrete lactate. Oxidative decarboxylation of pyruvate. Pyruvate, formed during glycolysis, is transported from cytosol to mitochondrial matrix. 3-carbon containing pyruvate is converted into carbon dioxide and 2-carbon acetyl coenzyme A in the presence of coenzyme A, and this reaction is catalyzed by pyruvate dehydrogenase. Coenzyme A acts as a acyl group carrier in several metabolic reactions. This is extremely exergonic and irreversible reaction. Acetyl coenzyme A is also involved in oxidation of fatty acids. This compound acts as an intermediate in many biosynthetic reactions and is always oxidized to CO2 through carbon dioxide through citric acid cycle. Now, the citric acid cycle, it is also known as Krebs cycle and TCA or tricarboxylic acid cycle. It occurs in mitochondrial matrix. It consists of 9 sequential reactions. It starts with acetyl coenzyme A, combines with oxaloacetate to form citric acid. This is the first stable compound formed in this cycle, hence named as citric acid cycle. Oxidative decarboxylation takes place with the release of carbon dioxide and reduction of NAD plus to NADH. Isocitrate converts into an alpha ketoglutarate, changes into succinyl coenzyme A is converted into succinate by the hydrolysis of thioester bond and release of GTP by substrate level phosphorylation. FAG reduced to FADH2 by addition of 2 electrons and 2 hydrogen. The enzyme used in this reaction is succinate dehydrogenase, is not present in the mitochondrial matrix like other enzymes involved in the cycle. It is component of integral membrane protein present in inner mitochondrial membrane, facing its active site towards the matrix. NAD plus is also reduced to NADH2 during the conversion of malate to oxaloacetate. Now, the oxaloacetate is further ready for the condensation with acetyl coenzyme A in next cycle. There is a release of 2 molecules of CO2, 2 molecules of NADH2, 1 molecule of GTP and 1 molecule of FADH2 in this cycle. For the metabolism of 2 molecules of acetyl coenzyme A generated from 1 molecule of glucose, this cycle takes place 2 times. Now, the malate aspartate shuttle system. In the cytosol, NADH is oxidized to NAD plus used in glycolysis and in matrix. It is reduced to NADH, which can further used to generate ATP. We can break down this shuttle into 7 steps. Step 1. The NADH produced in glycolysis is used to reduce oxaloacetate into malate. Step 2. The malate then moves into the intermembrane space and then enters the matrix via an antiporter transport system. Then enters the matrix via an antiporter transport system in exchange for an alpha ketoglutarate. Step 3. In the matrix, the malate is then oxidized back into oxaloacetate and the pair of electrons are collected by NAD plus to form an NADH molecule. This NADH can now be used by complex 1 of the electron transport chain. Step 4. The oxaloacetate cannot move across the inner mitochondrial membrane, and so a transamination reaction is needed to convert it into aspartate. Step 5. The aspartate can now flow out of the inner membrane via an antiporter system in exchange for glutamate. Step 6. The glutamate that moves into the matrix transfers an amino group onto oxaloacetate to form aspartate and alpha ketoglutarate. Step 7. The aspartate transported into the cytoplasm is deaminated to form oxaloacetate. The amino group is used to form glutamate from alpha ketoglutarate. Now, the electron transport system. The process in which electrons are transferred from NADH and FADH to oxygen and the energy released in this oxidation reduction reaction is used to synthesize ATP from ADP is known as oxidative phosphorylation. This is high energy yielding reaction occur at inner mitochondrial membrane through a series of complexes. Movement of electrons via an array of electron carriers is integrated with translocation of protons from the mitochondrial matrix to the intermembrane space. Movement of protons builds the proton motive force and is responsible for ATP synthesis. There are 4 complexes through which electrons pass to oxygen and several metal ions or prosthetic groups which are compactly and specifically connected with these complexes. In electron transport chain, electrons enter in chain from NADH, move from complex 1 to complex 3 and then to complex 4 by passing complex 2. The electrons which enter from citric acid intermediate succinate are transferred to FADH2, then to complex 3 and 4 bypassing complex 1. Complex 1, which is NADH coenzyme Q reductase, electrons from NADH enter into the chain and transfer to coenzyme Q by this complex, which consists of 40 polypeptide chains. Firstly, electrons are transferred from NADH to FMN, a cofactor related to FAD, and afterward through FES carrier. Finally, to coenzyme Q. There is a drop in electric potential by minus 360 millivolt. There is a drop in the electric potential by minus 360 millivolt and the energy released is minus 16.6 kilocalorie per mole. Coenzyme Q is also known as ubiquinone and acts as carrier which transports electron from complex 1 to complex 3. It is the only prosthetic group which is not protein bound. The oxidized quinone from coenzyme Q can accept electron to form semi-quinone. Coenzyme Q accepts electrons released from complex 1 or complex 2 and donates to complex 3. Movement of each pair of electron by coenzyme Q is linked with transport of movement of each pair of electron by coenzyme Q is linked with transport of 2 protons from the matrix in the intermembrane space fluid. Succinate coenzyme Q reductase. Succinate coenzyme Q reductase, which forms a complex 2, is a distinct protein complex consists of 4 polypeptides. Succinate coenzyme Q reductase, which forms a complex 2, is a distinct protein complex consists of 4 polypeptides. Succinate dehydrogenase is one of the 4 subunits of the complex 2 involved in citric acid cycle in the conversion of succinate to fumarate. The 2 electrons released in this conversion are transported to FAD and then to FES cluster and then to coenzyme Q. Finally, from coenzyme Q to complex 3 and complex 4. Coenzyme QH2 cytochrome C reductase, which forms a complex 3, consists of 10 polypeptides, and in this electrons are transferred from cytochrome B to cytochrome C. This is an energy yielding reaction that generates minus 10.1 kilocalorie per mole. Cytochrome C is a water soluble peripheral protein diffuses into intermembrane space. Both cytochrome C and coenzyme Q act as mobile electron shuttles and transfer electrons between the complexes. Cytochrome C oxidase that forms the complex 4, in this electrons are transferred from cytochrome C to cytochrome C oxidase after reduction of cytochrome C by complex 3. In complex 4, electrons are further transferred from copper ions to cytochrome A and A3 and then to the final acceptor oxygen and generates water molecules. 2 protons are translocated across the mitochondrial membrane during the transportation of each pair of electron through the complex 4. The structure of ATP synthase or F0-F1 complex. This complex has 2 components, F0 and F1. Both of these proteins are multimeric. F0 component consists of 3 integral membrane proteins A, B and C, 1 unit of A, 2 subunits of B and usually 10 subunits of C are organized as A1, B2, C10 in plane of ring and embedded into mitochondrial membrane. F1 portion is a water soluble complex of 5 polypeptides. Composition is alpha 3, beta 3, gamma, delta, epsilon. In this way, this channel is divided into 2 halves, half channel 1, half channel 2. Half channel 1 permits protons to travel one at a time from exoplasmic medium and attach to aspartate 61 in the middle of C subunit proximate to the center of the membrane. Half channel 2 allows proton to detach from the aspartate and transfer to cytosolic side. Alpha and beta subunits form a hexamer which is position the rod shaped gamma subunit which is fit into the C ring of F0. The epsilon subunit is formally attached to gamma subunits and delta subunits link one of the alpha subunits in the F1 complex to the B subunits of F0. As a result, the F0 A and B subunits and the F1 delta subunits and hexamer form a firm assembly fastened in the membrane. The binding change model. This is most acceptable model which explains the mechanism of ATP synthesis. This model supports the indirect coupling of proton motive force with the ATP synthesis. 3 beta subunits present in F0-F1 complex can attach to ADP and inorganic phosphate and catalyze the ATP synthesis. The downhill movement of protons through F0 moves C ring and gamma subunit of F1 particle. There are 3 conformation states of F1 complex by the movement of gamma subunit with the fixed hexamer subunit, which exposed the nucleotide binding sites at beta in following 3 states. The O state where ATP binds with beta very weakly. L state where ADP and inorganic phosphate binds with beta very strongly. T states in which the beta binds with ADP and inorganic phosphate and catalyzes the synthesis of ATP. It also binds very intensively with ATP. Then the final rotation of gamma subunits brings the beta subunit to O state and consequently releasing the ATP, and then the next cycle starts again. Alpha subunits also have ATP and ADP binding sites, but they only involve in allosteric regulation and not in ATP synthesis. Transport of ATP through inner mitochondrial membrane. Proton motive force is involved in ATP-ADP exchange mechanism transversely the inner mitochondrial membrane. Protons are required for further initiation of oxidative phosphorylation in the cytosol. This exchange is mediated by PMF, which involves phosphate transporter and ATP ADP antiporter, through which one proton flow from inner mitochondrial membrane to cytoplasm. Proton translocated during electron transport are accessible for ATP synthesis. It has been assessed that for every 4 proton translocated out, 3 protons are responsible for exchange and 1 is for synthesis of ATP. Now, fat metabolism. Oxidation of fatty acids occur in mitochondria. Fatty acids are both oxidized to acetyl coenzyme A and synthesized from acetyl coenzyme A. Fatty acids are converted to an active intermediate prior entering into the mitochondria. This step requires ATP and coenzyme A catalyzed by acyl coenzyme A synthetase, which is also called thiokinase, results in the formation of acyl coenzyme A or an active fatty acid AMP and pyrophosphate. However, long chain acyl coenzyme A cannot enter the inner mitochondrial membrane, so carnitine palmitoyl transferase 1 located in the outer membrane catalyzes the conversion of long chain acyl coenzyme A to acyl carnitine, which can penetrate the inner mitochondrial membrane. Carnitine acyl carnitine translocase serves as an inner membrane exchange transporter. Acyl carnitine is moved in coupled with the moving out of one molecule of carnitine. The acyl carnitine then binds with coenzyme A catalyzed by carnitine palmitoyl transferase 2 present in the inside of the inner mitochondrial membrane. Acyl coenzyme A is renewed in the mitochondrial matrix and carnitine is released. In beta oxidation, 2 carbon atoms at a time are cleaved from acyl coenzyme A molecule beginning at the carboxyl end. The 2 carbon units produced are acetyl coenzyme A, thus palmitoyl coenzyme A forms 8 acetyl coenzyme A molecules. The primary step in the process is the elimination of 2 hydrogen atoms from the second and third carbon atom catalyzed by acyl coenzyme A dehydrogenase and requiring FAT. This results in the formation of enoyl coenzyme A and FADH2. The reoxidation of FADH2 respiratory chain necessitates the mediation of additional flavoprotein. Water is needed to saturate the double bond and form 3-hydroxyl coenzyme A catalyzed by enoyl coenzyme A hydratase, which undergoes hydrogenation on the 3 carbon catalyzed by 3-hydroxyacyl coenzyme A dehydrogenase to form the 3-keto-acyl coenzyme A. Lastly, it breaks at the second and third position by thiolase forming acetyl coenzyme A and a new acyl coenzyme A, 2 carbon shorter than original acyl coenzyme A molecule. The acetyl coenzyme A produced by beta oxidation of fatty acid is further metabolized via citric acid cycle in mitochondrial matrix. Now, last is the summary. Glucose and fatty acids are the products of photosynthesis in the plants, and this process consumes energy, while the oxidation of glucose and fatty acids through cellular respiration produce huge amount of energy in the form of ATP. This ATP production occurs at different steps of glucose oxidation like glycolysis, Krebs cycle and etc. FADH and NADH are produced, which produce ATP by generating proton motive force. Oxidation of fatty acid produce more energy than glucose. This ATP production occurs at different steps of glucose oxidation like glycolysis, Krebs cycle and electron transport chain. FADH and NADH are produced, which produce ATP by generating electron motive force. Oxidation of fatty acids produce more energy than glucose. Thank you.